Hello everyone. Welcome to the BioC 2021 Career Panel session, where we will talk about possible career paths involving bioinformatics. And before we start, as usual, just a couple of housekeeping items. So if you have questions for the panelists, please enter them in the Q&A tab. And in case they are for a particular person or a particular career path, please remember to indicate that so that they will be directed to the right person. They can also be general, of course. And you can also upvote questions, and I think you have already noticed the emojis that you can use to send feedback as well. So I think that's it. Without further ado, I will hand over to Jenny Durnovich, who will guide us through this session.
Thank you, Charlotte. Let me share my screen here. So I put in the chat a link to my very few slides that I have. It's coming up here, but if anybody would want them. So this is the Career Information Panel. We decided to put this together for ideally aimed at graduate students and postdocs, but there are many of us who change career paths, so it would be good for you. So what are some of the possible career paths involved in bioinformatics? And so what is bioinformatics? A lot of people can't agree on exactly what it is. It's a relatively really new field, emerging only last maybe 25, 30 years if the word has been around. The main thing that it needs is biology. So I stole this nice Venn diagram from this blog post over here, and it's also a really great blog post about what is bioinformatics and what kind of careers that you can do in it. I mean, it's so good, I almost felt like I could just point you to it and then we don't have to have this panel. But anyway, back to here. Biology kind of does form the main core of it. We're doing work on biological questions. And then the two other main areas are statistics and computer science. And so in this figure, bioinformatics is, the intersection of all three. I'll be generous, and if you're doing any of these other two, biostatistics and or computational biology, welcome, you're in the right place. So there are many areas within bioinformatics, but there are also from that, there are many different ways that you can go. And so most people come into bioinformatics probably primarily from one of those three different areas, either biology or computer science or statistics. Then they pick up at least one more of the area, and they're in a traditional academic. And so the first career paths they think about or they see are those of that. So you've got your tenure, your non-tenure track professors, there can be researchers or other instructors. So there's a lot of different things that can be done in there. And the main hallmark, I would say, of these is that these individuals mainly work on their own projects. They will have a handful of collaborations with other people that need bioinformatics help and support. But so many people, so many biologists need bioinformatics support that there's been an outgrowth of what we call core personnel. And actually, we realize this also grows out of many places will have lab cores. So they'll have sequencing machines and they'll have expertise in lab software and other things. So they also have it for core personnel. So these are usually within a university, research hospitals. I've seen some independent consulting businesses where all they do is help people analyze their data. And the defining trait of these are we are mainly providing bioinformatics support for many, many other researchers. Now, kind of the third area, which always gets carved off, is industry. The main defining character is you work for a private or publicly traded company, but other than that, they can probably do any of these jobs above. I don't know that much about industry. We have some panelists from industry, so hopefully they will expound upon that more. And then also I would want to say that these careers, we tend to also traditionally think of them in sort of the Western world, but there are lots of opportunities around the world and the other developing nations, and we have a panel that hopefully Leo can speak to that about other places to work. So these are our panelists. I'm not going to introduce everybody. They're going to introduce themselves, but this is going to be the order. I put it on in case you want to contact somebody else later. And I want all the panelists to answer these three questions in just 3 minutes sort of thing. Describe your current position and how you got there. What is your favorite and least favorite part of your job? And what do you wish you knew as a graduate student in terms of your career now that you're doing in bioinformatics? Okay, so I will start out. And then after this, once we get through all the introductions, hopefully we'll have plenty of time for questions that you can put to the panelists. Okay, well, I'm an analyst at the Roy J. Carver Biotechnology Center at the University of Illinois, and this is a core service center. So we have a sequencing center, flow cytometry, proteomics, metabolomics, and we provide bioinformatics core with that. So I do the bioinformatics and statistics, approximately, 25 to 30 projects per year, mainly gene expression of one sort or another. I do some isolation, some microbial analyses, and I help out on some other things. I also help teach workshops for our researchers here about 6 times a year. Now, I come in from a completely biology point of view. My PhD is in animal behavior and evolution. I at least did have a really good statistics component to that, and then I switched into the postdoc in quantitative genetics, so more statistics. And this was right at the beginning of the advent of microarrays. And so I was the first user of the Affymetric system at the University of Illinois and the Core Service Center. And then I had been on the academic job market for 2 years and decided that wasn't for me. And I saw that they had a job posting for an analyst on a Thursday afternoon. It closed on Friday. I got my application in. They called me on Monday. I interviewed on Wednesday, and they gave me the job offer like the next Monday. So less than 2 weeks, which is such a difference than the academic job market. So that's that. My favorite part of my job is that I get to work on so many different types of biological questions and species. We're a land grant. I do pig, cow, horses, dogs, cats. We do human. We got cell work. I've worked on, my favorite one ever was the strain of yeast that is specific to make tequila. Other part of my favorite job, I don't have to be the primary author of grants and papers. Now, the least favorite part of my job is having to work on multiple projects at the same time, and then everyone has an urgent deadline next week. And also, because I'm helping everybody else, I don't have free time to explore the things that I find interesting. And what I wish I knew as a graduate student, I should have taken some computer science and programming courses. That's where I feel like I'm weakest. That's where my imposter syndrome comes up. I mean, I have a PhD in biology. I've got many graduate level classes in statistics, and I took a basic programming class in the 4th grade, and that was it. Okay, so I will move on to next to Haley Bellai. I don't know if you do want to just talk.
Yeah, I don't have any slides, unfortunately. But hi, my name's Haley Bellai. I'm an assistant professor at New York University Grossman School of Medicine, so kind of a classic academic track position at a large research institution. So a lot of my duties are writing grants, publishing, and I teach a class or 2 each year. So my road to this position was a bit untraditional. So I'm in a biostatistics division in the med school. I actually don't have a degree in statistics. My PhD is actually in engineering. So it's never too late to switch career paths, even if you're deep into your PhD. But basically, I worked in a classic wet lab, neuroscience lab. Basically, was posed with the situation of having to analyze a bunch of my data, and I had no idea how. So I started taking statistics classes on the side and realized that was actually my true love. So basically, I then went on to do a postdoc in biostatistics to get more formal training while taking statistics classes during my engineering PhD. And somehow it all managed to work out. In terms of favorite and least favorite part of my job, I truly love my job, which I know a lot of people can't say that. I tell people I'd give it a 9 out of 10. And really the only reason for that doc is just sometimes it is a lot of work. There's sometimes, I often do work almost every day. Sometimes you're like, gosh, I really just want to quit at 5 p.m. on Friday and start again at 9 a.m. on Monday. And sometimes you just can't do that, especially during the COVID pandemic right now. But overall, I just get to work on so many cool projects. I'm fortunate to have an amazing chair. Really great collaborators, and I get to pick on what I work on, which is just really exciting. In terms of advice for what I wish I knew as a graduate student, I wish somebody would have told me, don't stress out and lose sleep and, all these things that, we've all been there, and you have these moments of like, what am I doing with my life? And is this all going to work out? It is like, don't forget that, even if you don't get your dream job, you are super qualified. You're a PhD student in a STEM discipline, or even a graduate student in a STEM discipline. Like, the world wants you, and like, trust me. So it is all going to work out. And also the advice that Jenny gave, I mean, like, it's important to like take advantage of this opportunity to just learn. Like, when else in life are you paid, especially if you're a PhD student, to learn and to train? And don't be hard on yourself if you don't know things. Like, you're there to learn. So I wish, you know, even though I was taking stats classes on the side, you know, now I wish I had taken even more courses in like machine learning and other disciplines that I think would now be helpful with certain projects. So I think that's 3 minutes. So I'll pass on to the next person.
Okay, Katie?
Hi, my name is Katie Campbell. I'm a director of data science at Novartis Institutes of Biomedical Research. So I guess to describe my current position, I lead a team that's really interested in analyzing high-dimensional biomarker data from clinical trial subjects. So Novartis is a pharmaceutical company, and we run lots of clinical trials. Our focus is oncology for my group, and for those subjects enrolled in our clinical trials, we collect samples from them so that we can understand their response and sort of develop better drugs and better trials in the future. Most of the data we analyze is omics type data. In terms of my favorite part of my job, I really love talking to people and working on interesting scientific questions that really have an impact, translational impact in patients, really thinking about focusing on what that impact is for teams and making that happen. My least favorite part of my job, I work for a really big company, and it's sometimes hard to figure out who the right person to talk to is and how to make the right connections. So I think that part can be challenging at times. What I wish I knew as a grad student, I think a lot of what's already been said is certainly relevant. I think, you know, definitely encourage to ask questions and explore what other people are doing to learn as much as you can. Taking programming classes, I certainly wish I would have had more formal training, sort of picked it up as a postdoc. And I guess the other thing I would say, that the best advice I got when I was a grad student was, when you get to the point, or a postdoc or sort of wherever, when you get to the point where you stopped learning, it's time to graduate or transition from your postdoc if you're, you know, in a training role. So I would definitely pass that advice along to people.
Next up is Leo.
Sorry, my computer is a bit slow, so I keep unmuting myself, and I think the icon doesn't update. So I have a little slide here about my history, how I got here, but basically, I can share. But the basic story is I've alternated a bit through what you call academia and industry. After my undergrad, I worked 2 years at a startup company in Mexico. That's where I was like, okay, I want to like learn more about R and Bioconductor. I went back to grad school. After that, I joined a research institute that is a nonprofit institution in the U.S. It was kind of like a postdoc type of job, but like, but I also had like other responsibilities beyond the regular postdoc. So the challenge there was how to stay like relevant in academia in terms of publishing and things like that, while taking advantages of the opportunities I had there. Right now I'm an investigator at a place called the Lieber Institute for Brain Development, and one of the other questions you had was what are the things I wish I knew as a student, right? And so I would say there's three things there. One of them is like don't postpone your happiness type of thing. Like you'll always be stressed, you'll always have things to do, right? One thing I didn't know, but I guess I've realized more recently how important it is like the people you meet early on in your career can have a huge influence, right? So, for example, Davide right here, who's one of the next speakers, we met like at one of these YSC conferences years ago when we were just getting our careers started. And like another undergrad student from Australia, David McCarthy, he was instrumental in me like learning how to write like cover letters in English, right? So my first language is Spanish. The least favorite and least favorite parts of my job. The favorite is working with like some really like motivated people that think a lot about what we want to do and why we want to do it. The least favorite part of my job, but it's actually part of my job, is like dealing with people that like maybe don't have the right background to say like this thing that I'm asking is gonna take like X amount of time. So they want it ready for tomorrow, and you're like, well, that's not how this works, right? So it's not like I don't know if you've seen this meme about push the button bioinformatics. It's not like that. So my job is part of my job is trying to educate those people too, right? But dealing with stress is not my favorite. Thank you.
Okay, so then that next on the list is Jim McDonald.
Hi, my name is Jim McDonald. My current position is I work for a core facility at the University of Washington, and I got there. So I graduated with a bachelor's in microbiology back in 1985, and after a few years in industry, I went back in 2000 and got my master's in biostat, which I'm sort of non-traditional in as much as Jenny is, where kind of a biologist that sort of learned statistics late in life. Well, she wasn't late in life, but I was. And so after doing my master's at University of Michigan, part of the work that I was doing there was I was working for a core facility that was doing these new things called microarrays back in 2000 when they were new. And it was one of those things where it was a wet lab where they would run the microarray and then they would basically generate the data and then give it to the biologist to say, well, there you go. And the biologist would say, okay, so what do I do now? And so as my RA, as a master's student, I worked in that core facility analyzing the data for people. And after I graduated, they said, hey, do you want to stay on? So my getting my first job was even easier than Jenny's. They basically said, don't leave. And in the intervening period, so when I was working at that core facility, we were working with these Affy arrays. And that was back when Bioconductor first started. And they didn't have CDF environments for all of the Affy arrays. And so I was generating those. And I would email the Bioconductor people and say, hey, do you want this? I just generated this thing. Do you want to like put it on your website? And after doing that about 30 times, they said, hey, do you want to join Bioconductor? And in the intervening period, I went to a couple different labs, and then I moved to Washington, and here I am. So the favorite and least favorite part of my job, the favorite part is probably that I get exposed to a lot of different things. I mean, most of the things that we're working with are environmental and occupational exposures. So I guess it's sort of cool in some respect and not that cool in other respects. I mean, some of the things that people are exposed to. So getting to see lots of different stuff is really cool. And so dealing with a lot of people is good. Dealing with some people is not so good. I don't get to choose who comes through the door. I don't get to choose where they got their data, how they generated it. You know, ostensibly they come to us first and they say, hey, we want to do this thing. How should we do it? And that doesn't always happen. So sometimes I'm in the unenviable position of telling people what their experiment died from instead of helping them analyze the data. So that's probably the part that I like the least. And what do I wish I knew as a graduate student? So, as I said, I got my graduate degree at the University of Michigan, which likes to think of itself as being one of the top departments in the country. And so the whole how do you analyze your data part using computers is something that they expect you're just going to pick up on your own. So you learn all of the theoretical aspects and the sigmas and the betas and the omegas and things like that. But as far as how do you like start R and do things is sort of something that is evidently a trivial thing. So I wish I'd known that really the main part of my job would be knowing how to use R and figuring out how to learn about new packages and how do you read help pages and all that. And that's something that, you know, my work with the support site, I'm trying to sort of pay that forward because I've been doing that for 20 years now and it's not trivial. And I think that helping people figure that out is probably something that's useful.
All right. Thank you. Now we've got Russell Boehner.
Hi, I'm Russell Boehner. I'm head of computational biology at Maze Therapeutics. So to give you some background, Maze Therapeutics is a small biotechnology company that is focused on trying to figure out better ways to use human genetics information to identify core mechanisms of disease biology and basically improve drug discovery. The way we do that is by cultivating a pretty top-flight human genetics and functional genomics core that works very closely together to sort of rapidly nominate targets and then perform a series of numerically informed and genomic, computational, et cetera, analyses to kind of quickly vet the underlying theory of the disease biology and identify targets, and then move that on to the chemistry wing of the organization. And so you can probably imagine that the data science team, and specifically the computational biology team, plays a pretty core part in essentially harmonizing all that information flow. And also, luckily, we were able to work kind of as co-equal scientists with those different organizations to help them design their practices and analyses and interpret their data. So that's mostly what I do. How I got there was very similar path to what everybody else has described. And I think that kind of connects nicely to the answer of to what I'd like to say about what I wish I knew about as a graduate student. So I think one of the things that I notice for many of the panelists is there's an emphasis on this kind of serendipitous nature of, as Jenny says, landing in a field that basically just started about 25 years ago. And, you know, at the time when, you know, I was doing microarrays in the lab, it wasn't really even clear that that was separate from biology or molecular biology, or that that would turn into a new thing, I think. And so what I'd like to kind of suggest to people in graduate school is that there's this interesting connection between the credentialing that you get in your explicit training. So that would be things like undergraduate, graduate school, postdoc, early industry, that sort of thing. And the thing that you actually get hired for, which is not usually your credentialing. So you actually get hired for your skills, which are related to your credentialing in some ways, or hopefully more related to your credentialing, but they also are embedded in this kind of broader skill set that you need to develop in a kind of more active way. So these are things like written and communication skills and things like that that people talk about in a graduate training program, but I think it also involves a lot of just developing yourself as an interesting person who's interested in lots of different things. And you can prepare yourself by being curious about how various aspects of chemistry work, or various aspects of mathematics or statistics work to be in a position to take advantage of an awesome new career that develops during your lifetime, and push into it. So that's kind of my recommendation. In terms of the things that I like and I don't like about my job, I was thinking about this, and this is a surprisingly hard question for me because I think many of the panelists, I really like my job in most ways. What I would like to emphasize, though, is that I think what motivates me very deeply, and what I suspect motivates many or maybe all scientists, is this kind of underlying desire to do things better. So that could be either improving on an existing method for understanding a particular assay or technology. It could be about finding a better way to derive insight from a complicated experiment, or it could be things like just making a team work better, or understanding like a better system for checking in your code and keeping everyone on the same page about a large-scale team. And so the things that I enjoy and the things that I don't enjoy about my job are basically about how closely what I'm doing aligns with improvement and creating a better environment for my colleagues to work in, versus things where I feel like I'm kind of bound by historical precedent and requirements of the timeframes and things like that. That's all.
All right, thank you. Okay, two more. Jared.
Hi everyone. So I'm a bioinformatics research scientist at St. Jude Children's Research Hospital. I work in the Department of Developmental Neurobiology, mostly with the lab of Susie Baker working on high-grade gliomas. So I actually work with a few different labs, but she's my direct PI, and basically any collaborations that we do, I also handle all of those analyses. So how I got here was I'm actually pretty new. I earned my PhD just last year in June from Washington University in St. Louis, where I did a sort of damp PhD, is what we like to call it. So it was a mix of wet lab and dry lab work, studying lymphomas. So I came into grad school not really aiming to do computational things, but ended up generating a ton of data, and someone had to analyze it. So really delved into that skill set. Bioconductor was a huge part of that. And the more and more I delved into it, the more I really fell in love with it and enjoyed it. So that was sort of how I got here. St. Jude is, in most research hospitals, are kind of a unique mix between the academic environment and having more of a sort of industry-like feel to them. So it really, checked a lot of boxes for me. In terms of my favorite and least favorite part of the job, I'll really mirror a lot of what everyone else has said. I'll say my least favorite part is data cleaning, which, a lot of people will say you don't really enjoy bioinformatics, or you're not really, going to be a bioinformaticist if you don't enjoy that. But it's still frustrating, especially when you're working with lots of different groups, and organizational schemes are kind of all over the place. My favorite part is kind of a double-edged sword as well. I get to work on a lot of different projects from all sorts of different model systems and different diseases, and the underlying biology is very interesting. But on the other side of that, the task switching can really be a killer, especially when you're working with new data types and new systems that you're not familiar with. So I think prioritization and really knowing what's most urgent, and being on a clear page with everyone that you work with, and trying to be realistic with your estimates of how long things are going to take is pretty important. As far as what I wish I knew as a graduate student, basically what everyone else said. Imposter syndrome, everyone has it. It doesn't matter how good you are. It doesn't matter probably how experienced you are. I think, to some degree, everyone is going to run into that. And just, relax. It will be okay. Even when things seem terrible, science is mostly failure. And that's true for pretty much everyone. There's lots of things that don't work. It'll be all right. As far as the skill set goes, I think you do have to kind of take your training into your own hands sometimes. Make the effort to, go through and actually learn how to do analyses. Formal training can be helpful, but I think mostly you learn by doing. So really, take the time to learn things. You're never going to be in a better position than you are right now to, pick up some of these skill sets. Take the time to do it. Learn how to analyze your own data. read as much as you can, talk to people as much as you can, figure out how other people analyze their data. Really, try to always ask questions and be curious. And I've gone through the process of trying to find a position during the pandemic, which I think is a little bit unique. So if anyone has questions or anything, feel free to reach out to me. I'm more than happy to talk about that process. That's all I've got.
Great. Thank you so much, Jared. And last but not least is Davide. And I love saying your name.
Yeah, thanks. That was perfect. It's not every day that they pronounce my name correctly. But so yeah, my name is Davide Arista. I'm a freshly promoted associate professor at the University of Padua in the Department of Statistics. So how I got here is basically I got my PhD in statistics from the University of Padua, and then, which is in Italy. And then I moved to the US for my postdoc, and I did quite a long postdoc for stats standards, which was 5 years. And then I moved to the East Coast, and I got a tenure track position at Weill Cornell Medicine, which was a position very similar to Haley's, so biostats division in a medical school. And then about 2 years in, I got this offer from Padua to come back as a faculty, and it was just too good to say no. So I left my tenure track position at Cornell and came here. And so yeah, well, I guess my favorite part of the job is that I get to do what I love to do, right? So I love bioinformatics. I really think about that diagram that Jenny showed as what represents me and my research. I very much enjoy biology and stats and computer science, and bioinformatics is really what I want to do. This is hard sometimes in a stats department, which I guess it's the least favorite part of my job, is that especially if you're in a tenure track in a stats department, you're supposed to publish in Biometrica and these like very theoretical stats journals, and that's not what I like to do. So I made the decision early on to just do what I wanted to do as my research, and not just like do a classic stats path. But that's not always easy in a stats department. And I guess in terms of advice for grad students, well, on top of all the other advices that were really, really good, I think my advice is to reach out to people. I think I learned that mentors are very important, and finding a good mentors or good mentors is very important for your career. And your mentor does not need to be in your department or even in your city. Especially nowadays, you can just reach out to people, and most people are actually happy to kind of like give advice and mentor you. And I think this goes back to what Leo was saying as our first BioC meeting. I was not like very, I was not comfortable reaching out to people. I was kind of shy and sort of on my own. But like that conference, seeing like Robert Gentleman and like just approaching me and talking to me, I realized that it's actually easier than I thought to kind of like interact with people that you might put on a pedestal from the outside. And I think we talked about it with Leo many times. Like as the next generation of like bioconductor members, we really want to be what Robert and Rafa and Sandrine were for us. Like that I think is. Oh, and also I think I am the only panelist outside of the US. So I think it would be good for if you want some kind of perspective of like Europe versus US. I saw both words.
Great. Okay, so we have some in the chat and some of their questions. So let's see, since I can show this on stage, I'll do this one first. So for Jared Andrews.
Definitely, for sure. And feel free to reach out to me. There are a ton of open bioinformatics positions here. both at the staff level, the faculty level, and postdoc level. So definitely reach out to me. You can send me a chat on here, and we can get email addresses. My email address is jared.andrews@stjude.org. So feel free to email me. I can definitely put you in contact with some positions that would likely be a good fit for you. And that goes for anyone else as well. St. Jude is booming computationally, so yeah, just reach out.
Yes, one thing I would say as far as the job market, I know we can't hire people. We have jobs for people. We can't hire. Most people need somebody who's got bioinformatics, and we have a hard time hiring the people. Okay, so I'll show this one on stage as a general question. Would you recommend to go for a PhD if the final goal is to work as like a core analyst? Yes, no. I don't know. Jim, what do you think?
As a non-PhD, I say no. So it really depends on what your expectations are. So as a master's guy, I'm not really expected to publish or to generate new methods or things like that. So if your interest is in sort of higher level things like generating new methods and things like that, then you really do need to get your PhD. But if you're going to end up doing sort of what I do, which is, knowing how to put data into R and analyze it and be smart enough about it that you know what models you should fit and that sort of thing, I'm not so sure that you need a PhD for that.
Okay, so I will go back. There was one from Royet in the chats, said that now most of the companies have standard data processing pipelines in place that require minimum intervention, say RNA-seq data analysis and DEG. What are your thoughts on the future of bioinformatician as a core personnel? Because people really need more bioinformatician, and they think they have pipelines set up. Well, you need somebody to maintain the pipeline because the tools change constantly, all the time. There is no one button push. That's what everybody wants. One button push, give my data analysis out. And I think there's always going to be a need for the person that can take it and interpret it. I mean, that's my sense of that believe me, I feel like I have amazing job security. They would cry if I told them I would leave. Basically, the type of thing it is. Anybody else?
I have fairly strong opinions about this, if you don't mind me.
Sure, go ahead.
I mean, my general feeling about this kind of a question is that it's an entirely understandable one, but in my mind, it's not very well formed because it reminds me a lot of lots of conversations that I've had with people about like, why would you optimize an assay? We can sequence this RNA sequencing experiment with 100 million reads per sample, and that's within our budget. So why would you try and figure out how to do RNA-seq more efficiently or faster or something like that? And the answer isn't really that like there's going to be some end state where we decide, okay, RNA sequencing is done. What's going to happen is that as you improve those methodologies, as you improve that standardization and technique, then new ideas come along and new applications become apparent and available that really, really do require the kind of specialized expertise that the people on this call have and which the people who are listening to us are developing. I mean, if you are in a company that just like says, well, we know how to sequence RNA and we can get count matrices and you're done and let's fire all the bioinformaticists, you should find a new job because that company is not thinking about the right way to use its scientific abilities. So, I mean, like, I think that it's not an endpoint where this is going to just become PCR and it's optimized, and people still work on PCR, by the way. It's going to be something that opens up more avenues for understanding core aspects of biology.
Okay. All right. Thanks. So we'll move on. This one got upvoted. Several panelists mentioned they wish they had taken programming classes as a grad student. Any specific areas, topics that you recommend learning early on? I don't know. For me, I would just say just a general computer science class would have been useful since I didn't have even that basic level. What about other people?
I'll personally say that I think having really good data visualization skills and knowing how to effectively communicate biological information into figures that are easily interpretable is pretty important. I don't think most classes are really going to get into that, but having practice doing that and working on real data sets that are messy and not perfect, and figuring out the best way to deal with those is also super valuable. So as much as you can, work on real data sets, rather than something that's just cherry picked and standard. I think a data structures and algorithms class would probably be helpful for a lot of people, myself included. That's something I wish I had, a little bit better formal knowledge of. But in general, a lot of comp sci topics are going to be somewhat less important for you. Writing the most optimal code is not always the goal of bioinformatics, especially when you're just doing analyses. So that's kind of my take on it personally.
Okay.
I'll jump in really quick, just add to that. I would take that lovely Venn diagram that was shown at the start and kind of go in each of the little subcores and be like, which area am I the weakest, or, maybe I'm not informed as much in this area, and then maybe just make sure you've got courses kind of covering each sector.
Okay, here's one. I had several upvotes. Graduating soon, I've been leaning more towards core industry. I'm not sure if this is imposter syndrome or not, but I still feel unprepared. Should I do a postdoc to pick up some more skills or learn on the job as a staff scientist? Anybody want to comment?
I'll comment. I would do the on-the-job training. Basically, you will learn way more working than you will taking courses, because when you're taking courses, like people have said already, you have these artificial data sets where it's sort of busy work, where you have to try to figure out how to do something with it, but you don't really care about it. If it's your job and you have to figure out you have something that you have to figure out how to do. So you have an inherent reason to want to learn, how do I get this thing to work? And I personally find that that's a lot easier to sort of pick up a skill when I have an actual reason to do it. And as far as the earlier question about other skills, I think knowing how to use any sort of Linux command line stuff will probably help your life a lot. So if you know sed or awk or even Perl or Python, things like that, where you can do things on a stream, for a lot of things, that's way faster than trying to do it in R. So having a sort of a broad-based computer skill set is a really good thing to do.
I can add to that too. I think, coming from industry, we tend to hire from both places, from both people who at the PhD level have done a postdoc and people are coming straight from grad school. I think it's really, if you sort of look at the roles you're interested in and you see skills that you don't have yet, then doing a postdoc or some other sort of training to build those skills to make yourself competitive makes a lot of sense. I think if you already have those skills, there's a lot of stuff you're not going to learn until you get to industry. And we're hiring for people we think are going to be creative, independent thinkers, and we're not expecting them to know the ins and outs of drug discovery or clinical trials when we hire them.
Okay, we've got lots of questions here. Try to get through them. Remote working might be here to stay. As employers, would you prefer to hire individuals who are willing to relocate, or is this not an important factor?
I have opinions about that. I have opinions about all these things, but I'm trying to be a good boy and not talk through everything. Yeah, I think this is a question that's come up a lot in my group and also in the broader data science group. My general sense, at least in my company, and I think in many of my peer companies in the Bay Area at least, is that there is a core component of analytical work that really does require face-to-face interaction. So that means, I mean, partially that reflects the structure of our company where, you know, we do work very closely with bench scientists and with human geneticists and things like that. And it's really, really hard to just be as effective over Zoom. That said, I think the pandemic has done a nice service of helping people recognize that that doesn't have to happen every single day of the week, and that you know, people are very likely to transition, I think, to a much more hybrid schedule where you have, you know, days in which there's lots of face-to-face interaction, lots of individual meetings where, you know, you stand in front of a whiteboard and hash things out, but then other days when you stay home and just really focus on your work. That's been our experience anyway.
I actually have opinions as well. I have worked from here since 2011. So I work for the University of Washington, and I've been to Seattle like, I don't know, 5 times since 2011. So for a lot of what we do, as long as you've got a reasonable internet connection and you can log into the server, I mean, most of the stuff's not done on your laptop anymore. So I've been logging into the server at UW for like, it's coming on 10 years now. And I don't think that it's necessarily the way of the future, but I certainly think that there's room in our industry for that sort of setup.
Okay, where's one? Oh, I had it and I lost it. All right, well, I'll do this one. Lots of them from Nitesh. There seems to be limits to what your career growth with a master's in academic bioinformatics. Is that the case in industry bioinformatics as well?
I'm happy to answer. I can say that it's been really interesting. When I started at Novartis, there was actually different titles depending on what degree you came into the company with. But recently, they've actually done away with that. So you can't tell by title who has a master's, bachelor's, PhD. It's really based on the level of experience of that person and what their role entails. And I think that does a really nice job of helping level the playing field here. So I think, you know, I can only speak for the company I work for, but I think that there's less limits these days. And I think people are seeing that it's really about your level of experience more than your title.
I would agree with that. However, my experience when I was at Genentech before here, exactly the same thing, where you know, people coming in with a master's were just inherently on a different track than people coming in with a PhD or a postdoc. I think the one thing that I would mention is, and as you say, that's changed considerably. One thing that I would suggest, though, is that it's not clear yet if that is a consequence of the extremely tight bioinformatics labor market, or if that's a kind of broader step change in the industry. My suspicion is that because everyone deeply needs bioinformaticists right now, that things like credentialing become less important than getting an excellent person in the chair.
Okay, so yeah, so this is a related question, that there are benefits doing just a master's versus PhD, or going past PhD and postdoc before going into industry. So it's mostly been interesting. I would, as far as core, because that's, you know, you can also go into core either way, I would say. And in our group, we have a mix of just some masters and some PhDs. When we hire, we say we prefer PhD, but the master's is required. And we've had some amazing people here who are masters, and then they leave, and we're sad that they're gone. So let's see if we have other ones. So try to get through all the questions that we have here. Here's a different. Having many years postdoc in the field of RNA, now moved into bioinformatics and NGS analysis, are there positions that could accept someone bridging wet lab, dry lab, or are they a pipe dream? I don't know if Debbie Davis gets into a little bit, because we didn't talk about the 10 simple rules, folks, yet. That this is the, you know, some people say beware about being this person.
Yeah, well, I mean, I can mention the time simple rules in a moment, but I have like this question made me think immediately a friend of mine who has exactly that position in a startup and in the Bay Area. So I think that job exists. I don't know how common it is, but like especially with single cell RNA-seq, I think a lot of people that perform the experiment know the system much better than like a general bioinformaticist that would come up and just look at the matrix. And having that person being able to talk to the bioinformaticist on the same ground is very important, at least in a startup where you don't have like 100 people, right? 100 employees. So yeah, I guess Jenny mentioned the 10 simple rules. So last year at BioC, spearheaded by Federico Marini, we created like a group of people sort of drafting a paper on the sort of similar to the 10 simple rule papers that you see in PLOS Comp Bio. And we're actually, I mean, not as fast as we wanted, but we have a draft now, like a rough draft. So we have a channel in the Slack, in the Bioconductor Slack. So I can post it here, but you're all very welcome to participate and either to get a perspective from people on like all these things, and also to contribute if you want to.
Okay, I know we're a little past our time, but we've got one more question. So for Russell Boehner, what other skills are valuable for working in a drug company? I'm graduating this spring and trying to create my resume.
Oh man, that's a good question. I think one thing to say is that drug companies are not created equal. There are lots of different companies that work different ways. And I guess the first thing that I would say is that you need to be nominally qualified for whatever position it is you're applying for. And that sounds sort of obvious, but I think that, you know, obviously in my group, I'm never going to hire somebody who doesn't know how to use a computer, for example. That said, the things that I think make a drug, and the things that I think making a candidate most attractive actually involve things like cross-functional skills. So really kind of piggybacking on the last question, when I was hired at Genentech, and honestly here as well in the computational biology department at Mays, I would say that having like direct experience with bench biology is one of the kind of top secondary skills that we look for in a good informaticist. And that's really just based on this this empirical observation that people who have the experience of doing experiments themselves understand where problems can arise, the way that experiments fail, and also can come up with good ideas about better designing experiments and capturing components of variation that aren't well understood, for example. So I think that you know, the position in which one actually gets a job to both work at the bench and work from a computer is relatively rare. But having that cross-functional skills and then being embedded in a bioinformatics company is pretty common. In terms of the broader skills, I think this was posted very early in the talk, so this might be about like the kind of fluffier ideas that I had during my little introductory section. I think that there's a huge amount of skills related to just caring about your colleagues and understanding what they're interested in and trying to figure out how you can help them think about their problem in a different way. That's a very weird kind of skill, and it comes from places that are not very characteristic sometimes, but honestly, that's the kind of unsatisfying answer I guess I would have.
Okay. There's one more came in, and I will try to get to it, but I do want to say, because we'll have to leave here soon, there are some, because we're going into poster session and there's a networking, we do have tables that are set up out in the lounge, one for kind of the 3 different areas, core, academic. I have some time, I can go sit at one and answer questions. I don't know if anybody else, if they would have some time, wouldn't mind sitting at a table.
I'm happy to as well.
Yeah, to just the discussions. Katie, did you have anything else to add about hiring in?
No, I think that covered it well. Thank you.
Okay. there's been one more, now I can't find it, so let's see. Just because I want to answer this one. Do you enjoy writing grant applications? Do you have to deal with it? I utterly hate it. And so I'm so glad that I don't have to be the primary person writing up a paper or a grant or anything. That's just me, because that's the hardest part, what I found of academics is, and so that's just my opinion. So other people I know love writing grants. I had a friend who, she wanted to become a professional grant writer. And I was like, are you crazy? Anybody else?
Yeah, I guess I can chime in from the academic standpoint. I mean, I don't mind it. I think it's different when you're the statistician or the bioinformaticist, and you're not necessarily writing the whole grant. You're just, for example, I write up the power calculation and the statistical analysis section. And I think in that case, you know, you talk to the PI, they tell you what they're trying to do, and then you translate it into stats language. So I think collaborative grants can actually be a lot of fun. And I think in any situation, you know, doing things alone is really hard. And if you've got a solid team, it's a lot easier. Also, it gets easier with time. So your first grant or two is rough. And then you start realizing, you know, it's just kind of like checking off boxes of what sections to include and components. So.
All right. Okay, I think we will end there. So thank you to all the panelists. Thank you so much for many of you last minute coming on when I asked you. I think we got a really great group here. So I was very, very pleased with it. Okay, enjoy the rest of your conference. Again, if you have any questions for any of us, we'll be around, and just feel free to come up to any of us, which is great. All right.
Thanks, everybody.